This technique can answer key questions in regulatory processes, such as which role protein dynamics and organization play in functional adaptation. The main advantage of this technique is that it can be done in living cells, thereby revealing protein dynamics on the single molecule level. Though this method was originally implemented to provide insights into the spatiotemporal organization of mitochondrial membrane proteins, it can also be applied for other organelles and other membrane proteins. Demonstrating the technique will be Timo Appelhans, a grad student from my lab.
After transfecting and preparing cell samples and the microscope according to the text protocol, mount the prepared sample with fluorescent beads in the sample holder between the PTFE ring and the red rubber ring. Start the microscope, all hardware components, and all software needed for microscopy. Use a fresh lint-free tissue sprayed with isopropanol to clean the objective lens and the bottom of the cover slip. Then place a droplet of immersion oil on the pupil of the objective lens. Next, place the sample holder with the bead sample onto the microscope stage so that the bottom of the cover slip contacts the oil. Then focus on the beads by using transmission light or a laser line. Adjust the power of the two excitation lasers to achieve similar signal density in the two fluorescence channels. Then search for an area with many distinct fluorescent signals.